in this section we will see about compliance and elastance. Basically what is elastance? So elastance is nothing but resistance to a stretch, that is called as elastance. So when you stretch a rubber band, it is not easy to stretch it, it offers some resistance, that is called as elastance. Here again in the lungs, it is elastic. So it has got its own recoiling forces that is acting on the lungs, and that is called as elastance. Now what is compliance? The effort with which the lungs are distensible, the distensibility is called as compliance. Okay. So in order to distend the lungs, we need to overcome the two important elastic forces. One is the recoiling forces of the thoracic wall, the other one is the recoiling forces of the lungs. Okay. So the thoracic wall usually tends to pop outwards. Okay. So it expands. This is provided by the rib cage muscles and the tendons. Whereas the elastic recoiling of the lungs is the tendency to collapse inwards. Okay, so they tend to collapse inwards inside the thoracic cavity. This is provided by the tissue forces, which include the smooth muscle tissue, elastic and collagen fibers which are present in the lung parenchyma. The other important force is the surface tension. Okay, so this surface tension is present within the alveoli. So we just saw how the ribs are popping outwards, whereas the lungs are tending to collapse inwards. Okay. So there occurs a point where this elastic recoil of the chest wall and the elastic recoil of the lungs, they balance each other. So at that balancing point, there occurs no airflow into the lungs and outside the lungs. Okay. So that is called as relaxation point. Now what is basically compliance? By definition, it is the distensibility. It is nothing but the change in the lung volume per unit change in the transpulmonary pressure. Okay. So it is given by the formula delta V by delta P, that is the change in the volume per unit change in the pressure, and it is represented in liters per centimeters of water. So if you measure the compliance of the lungs and the thoracic wall together, it comes to around 130 ml per centimeters of water. So if you want to expand the lungs, so you need to expand the lungs, the alveoli, the lung parenchyma, the next layer is the pleura, you have to expand the pleura, and then you have to expand the chest wall also. Okay, so it requires more effort. Whereas if you record the compliance of the lungs alone, it comes to around 220 ml, little higher than the compliance of lungs and thoracic wall put together. Okay, so it is 220 ml. This we can measure experimentally by taking the lungs separately, and you can record its stretchability. Okay. Here we can measure the compliance of the lung at a given volume, but there is no airflow. So there is no pressure component involved in the static compliance. Now how can we measure the static compliance? We can measure the pressures across a range of lung volumes. Say for example, the patient is asked to take incremental breaths from a spirometer, which gives a known volume of the air that is breathed. After inhaling a particular known volume, the a mouthpiece of the spirometer is usually attached with a manometric device that can measure the pressure. That mouthpiece will be shut down and the pressure can be measured. Okay. So for that volume, how much is the pressure? The next volume, increment of the volume, how much is the pressure? Like that, we can measure the incremental volumes during inspiration and the pressures associated with that. Okay. So similarly, you can also do it for deflation. So this is a function of the elastic recoil of the lungs alone, which is actually including the surface tension component also. Okay. So this is about the static compliance. There is another compliance called as dynamic compliance, wherein the compliance is measured during respiration, wherein it involves continuous pressure changes along with the volume changes. Okay. So it includes the pressure as well as the volume that is required to generate the required airflow into the lungs and outside the lungs. This is quite specific, but it is always less than the static compliance, because here it involves not only the lungs but also the chest wall. Okay. But the most specific compliance is this called by its name specific compliance. Okay, this is actually calculated one which is given by the formula compliance divided by the lung volume. It is more reliable and specific and also constant for a particular individual, sort of customized to a particular individual. Okay. So, how this compliance is measured? We measure the intrapleural pressure by inserting an intraesophageal balloon that is tipped with a manometric device. So, intraesophageal pressure will reflect the intrapleural pressure as well. We saw that before. Okay. So, every time the patient is inspiring from a spirometer with a known volume, we measure the pressure. Okay. We shut down the valve and mouthpiece and we measure the pressure. Okay. And similarly, during the next known volume, we measure the pressure. Like that, we measure a series of inspiratory pressures with the volumes, and similarly we can do it for during the deflation process, measuring the series of volumes during deflation along with the pressure. When we plot this pressure and the volume both during inspiration and expiration, we find a typical pattern of graph. This is that graph. So, if we record the intrapleural pressure, we can see during inspiration when the pressure becomes more negative, the volume also increases. Okay. Similarly, during deflation, when the pressure becomes less negative, the volume also decreases. This slope is called as a compliance. Okay. But if you can observe, the inspiration is lagging behind the expiration. That means, at any given point of pressure, say for example minus 5 centimeters of water, you can check the volume of air that is present during inspiration. It is roughly around maybe 125. Okay. Whereas at the same point, at the same pressure, pleural pressure during expiration, during deflation, look at the volume of the volume present in the lungs, it is almost 350. Okay. So, at any given point of pleural pressure, the volume of air that is present in that inspiratory phase is lesser when compared to that present in the expiratory phase. So, the compliance of the lung is more during the expiration. Okay. So, this lagging behind is called as hysteresis. Okay. This lagging behind, this is called as hysteresis. Hysteresis means to lag behind. Okay. So, this is the typical pressure volume curve when we measure the compliance. So, if you have noticed this compliance curve during inspiration as well as during expiration, it is not a straight line. Okay. The graph relationship between the pressure and the volume is not a straight line, it is curved. Okay. This is because the lung has to overcome certain resistance. Okay. So, this includes a viscous resistance as well as the airway resistance that the lungs has to overcome in order to get inflated and also during deflation. That is the reason for this curved pattern. Okay. Now, this graph explains about the relaxation pressure curve. Okay. So, here in the x-axis the intrapulmonary pressure is plotted. Okay. So, if you can see here, the intrapulmonary pressure is negative on the left side and positive on the right side. Okay. In the y-axis, the volume is plotted. Okay. In the left hand side, we can see that volume is represented as change from the resting volume. On the right side, we can see the absolute lung volumes are represented. Okay. Now, if you can observe, at 0 intrapulmonary pressure, the volume is also 0. That means, this is a volume which does not changes. Okay, and it is actually represented as end expiratory volume, which is representing the functional residual capacity. At this point, there is pressure is also 0 and the volume is also not changing, and this point is said to be the relaxation volume. That is, the functional residual capacity which is present at the end expiratory point is said to be the relaxation volume. Okay. If you can see that the intrapulmonary pressure becomes positive at greater lung volumes, whereas it becomes negative at lesser lung volumes. Okay. So, the slope of this relaxation pressure curve gives you the compliance of the lung or the respiratory system in total. Okay. So, on the right side you can see the maximal expiratory curve has brought about the compliance curve at a more positive pressures. Similarly, the maximal inspiratory curve which is present on the left side is brought about by the maximal inspiratory effort which is seen during the most negative intrapulmonary pressure. So, in this picture you can see the pressure volume curve. Here instead of intrapulmonary pressure it is given as transpulmonary pressure. So, as per definition of compliance, it is the change in the volume per unit change in the transpulmonary pressure. When the transpulmonary pressure, the volume also increases until it reaches its peak value of 6 liters. Okay. So, then it becomes a plateau. This is a normal resting compliance curve. Okay. But if you can see in the down curve, that is the curve, compliance curve for fibrosis, even though the transpulmonary pressure increases, the volume is not increasing. That means the lungs are resistant, it is not getting stretched because of the presence of this restriction as in the name of fibrosis. Okay. Similarly, when you see at this top curve for emphysema, wherein the lungs have lost its elasticity, it is already distended. So, even with a smaller transpulmonary pressure, the volume of the lungs is greater because there is no force that is resisting the inflation or resisting the stretchability. The elastance is lost. The same thing happens in cigarette smoking also. So, what are the factors determining compliance? Basically, these are the elastic forces of the lung tissue offered by the fibers of collagen and elastin which are present in the lung parenchyma. The other one is the surface tension. This surface tension is given by the fluid that is present in the inner walls of the alveoli. So, if you can see, the surface tension is always seen wherever there is an air fluid interface. So, in the lungs also there is an air fluid interface that is better explained in this graph. So, this is the lung which is air filled and this is the lung which is saline filled. If you measure the compliance of an air filled lung, you can see the inspiration is different from the expiration and there occurs a lag behind, that is hysteresis is seen. Whereas, if you can see the compliance that is recorded for a saline filled lung, it is almost the same. The same effort is put for inspiration and expiration. There is no lagging behind. Okay. So, this demonstrates that the effect of surface tension is greater in an air filled lung. So, the factors affecting compliance includes the lung volume. If the volume is more, the compliance is also greater. Okay. So, in this picture you can see in the situation 1 where you have got both right and the left lung, if you can measure the compliance, say with 5 centimeters of increase in the transpulmonary pressure, there occurred increment of 1 liter inside the lungs, then the compliance is said to be 1/5, it becomes 0.2. Okay. In the situation 2, wherein only one lung is present, the compliance is exactly the half, that is 0.1. In the third situation, where the lung is just 1/10 of the normal, you can see the compliance has drastically reduced to 0.02. So, if the lung volume is greater, the compliance is also greater. Okay. But if you can see the specific compliance which is calculated specific to the given lung volume, then it is almost the same. See, for example, in the situation 1, it is the 0.2, that is the lung compliance divided by 1 liter, it gives 0.2. Okay. So, in the situation 2 and 3, also the specific compliance becomes 0.2. Okay. So, this is more stable. Then the next factor that affects compliance is the respiratory phase. We just saw during deflation, during expiration, the volume of air that is present in the lungs is greater when compared during inspiration. So, the compliance is more during deflation. Gravity, yes, in standing posture at the apex, the compliance is low because the alveoli is already distended. So, it is much difficult to distend the alveoli further. So, if the surfactant is more, the compliance will also be more, and vice versa. We will see about this concept of surfactant in the next few slides. So these are the diseases that are associated with the altered compliance. Decreased compliance is seen in restrictive lung diseases including fibrosis, because this offers more resistance to the stretchability, so that causes decrease in the compliance. Pleural effusion, again it needs more pressure to distend the fluid, okay. Tension, pneumothorax, consolidation, atelectasis, all these are the conditions that are associated with decreased compliance, whereas increased compliance is seen in emphysema and cigarette smoking. Both the condition results in loss of elastic fibers, so it is very easy to distend the lung tissues, alveoli. Next, we will move on to the concept of alveolar surface tension and the surfactant. Have you ever wondered why the raindrop is globular or tear shaped? Similarly, the droplet of the water which is coming from the tap, why it is in a tear shaped or a globular shape? Have you wondered this? Have you ever seen a mosquito or any insect walking on the surface of the water? We know that insect is heavier than the water, but still it walks on the surface of water. This is all possible because of the concept of surface tension. Now, what is this surface tension? Now, whenever there is an air-water interface, what happens is there occurs the attraction between the molecules of the water, especially at this surface. Okay. So, this is what is happening. This is the air, this is the water phase. There are different molecules which are present in the water. Okay. This is the surface molecules. Okay. So, what happens, each and every molecule exerts some intermolecular attraction between each other. Okay. So, similarly, here again the surface molecules, they tend to have some attractive forces between each other and to the molecules downwards, but there is no molecule above this layer. So, what happens, all the forces, they tend to drag downwards. Okay. So, this exerts a pressure and that pressure is called as surface tension. We have the air-water interface in our alveoli too. Okay. So, if you can see, this is an alveoli. There is a thin layer of fluid which is present in the inner wall of the alveoli, and here is the air. So, there occurs air-fluid interface. Automatically, the fact of surface tension comes into play. So, this surface tension reduces the surface area in the alveoli and they tend to collapse the alveoli, causing collapse of the lungs as such. Okay. Okay. This guy Laplace put forth his law stating that in any sphere with a hollow inside, he said that the pressure developed will be equal to twice the surface tension divided by its radius. Okay. So, this pressure here in the alveoli, we refer it as collapsing pressure. In some other instance, we refer that as distending pressure. Here in the alveoli, we refer it to as collapsing pressure. Okay. So, this collapsing pressure is equal to twice the tension, surface tension over the radius. Okay. So, as per this equation, whenever the surface tension increases, the collapsing pressure also increases. Whenever the radius increases, the collapsing pressure decreases. Okay. So, what happens during expiration? The alveoli becomes deflated, it becomes smaller. Okay. So, the pressure becomes constant. If the tension is not reduced, what will happen? It will completely close. Okay. So, that causes complete closure of the alveoli and the lung collapse. Then now what will happen to the effort for the next inflation cycle? It becomes very difficult to open up the completely closed alveoli. So, that is the effect of surface tension. So, this is what is happening. Imagine there are two alveoli. This is a smaller alveoli. This is a relatively larger alveoli. This smaller alveoli has got a mucus plug that is obstructing. Okay. So, air can come inside with efforts. Okay. After that, during deflation, what happens? The surface tension will be acting and it causes the pressure to collapse the alveoli. Okay. So, with this high pressure, the air will get rushed out of the alveoli and moreover it gets diverted to this alveoli which is larger. Here the surface tension is also the same, but what happens, since the radius is larger, the distending pressure or the collapsing pressure will become lesser. So what happens? Because of the air also coming from the smaller alveoli and also the air which is coming from the atmosphere, this alveoli tends to grow larger and larger, whereas this alveoli tends to grow smaller and smaller. This causes instability of the alveoli. Okay, so there becomes different sizes of the alveoli. But this is not happening normally. So there is something in our normal lungs which prevents this collapsing of the alveoli and also prevents too much distension of the alveoli and provides stability to the alveoli. That is this surfactant. Okay. So this surfactant is actually a chemical which is a combination of proteins and the lipids. They are secreted from the type 2 alveolar epithelial cells. Remember, the alveoli, it has got a basement membrane and it has got two types of cells. One is a type 1 cell and the other one is a type 2 cell. We call it as pneumocytes as well. Okay. So this type 1 cell is predominantly involved in the gas exchange, whereas this type 2 cell is actually secreting this surfactant molecule. Okay. So the surfactants is produced in the type 2 cells, they get stored in the lamellar bodies and whenever needed, they get released by exocytosis and they also get removed by this pulmonary macrophages. So this is the composition of the surfactant. The chief component is this dipalmitoyl phosphatidylcholine, phosphatidylglycerol, other phospholipids are there, there are neutral lipids, proteins are there, surfactant proteins A, B, C and D are there, little bit of carbohydrates is also present. Okay. So if you can see here, this is the alveoli. The pink color is the alveolar membrane, the green color layer is the fluid and you can see the black color layer, that is the layer of surfactant. Okay. And beyond that you have got the air. So this surfactant prevents the air fluid interface here. If you take a closer look on this surfactant layer, you can see this is actually a phospholipid which is having a globular head and two tails. Okay. The head is hydrophilic in nature which is facing the fluid side, whereas the tail is hydrophobic in nature which is facing the air side. Okay. So what is the mechanism of this surfactant, how it prevents the surface tension? This alveoli is the alveoli seen at the end of expiration. So what happens, the surfactant molecules here, when the alveoli is smaller, it becomes thickly distributed, that is densely distributed. There is increase in the surfactant to alveolar surface area ratio. So what happens, it covers the entire alveoli here. So the surface tension effect to collapse the alveoli is prevented. So this prevents the complete closure of the alveoli during expiration. But what happens during end inspiration, the alveoli is now inflated, it is larger. So when it gets larger like a balloon, it blows up, the surfactant becomes widely distributed, it moves apart. Okay. So this also has got one important function. Because of this, the surface tension lowering is little bit lesser, but what happens, it actually prevents the over distension of the alveoli. So summarizing the physiological effects of the surfactant, surfactant reduces the tendency of the alveoli to collapse by reducing the surface tension to almost 7 to 14 times. If you consider the surface tension of a pure water molecule, it comes to around 70 dynes per centimeter square. So if you take the alveolar fluid without surfactant, it comes to about 50 dynes per centimeter. But with surfactant, the surface tension is reduced to just 5 to 30 dynes per centimeter square. Surfactant reduces the work of breathing. See, at the end of expiration, what happens? The alveoli are just only partially closed with the help of the surfactant. Okay? Without surfactant, it will be completely closed. So what happens during the next inflation of the same alveoli? It is much easier, only little transmural pressure is required to open up the partially closed alveoli when compared to the completely closed alveoli. So that is how it reduces the work of breathing. Surfactant helps to prevent the pulmonary edema. This is very, very important because surface tension is an inward drawing force. Okay. So it draws fluid, it is a retracting force which can draw fluid with the pressure of around 20 millimeters of mercury. So, with this force, some amount of the fluid which is present in the interstitium can also enter into the alveoli, resulting in the pulmonary edema. That is prevented with the help of the surfactant. Finally, because it stabilizes the alveolar size and makes it uniform, the gaseous exchange also happens effectively. These are the functions of the surfactant in our lungs. Now, these are the factors that affect the surfactant synthesis. Whenever the main bronchus is occluded or a main pulmonary artery is occluded, or because of cigarette smoking, or after inhalation of 100% oxygen for a long term, the surfactant secretion decreases. Okay? Whereas thyroid hormones and the steroid hormones increases the surfactant synthesis. Thyroid hormones causes increase in the number and size of the inclusion bodies which are present in the type 2 cells, whereas steroid hormones accelerate the maturation of the surfactant. Okay? Deficiency of this surfactant happens in preterm babies, is a very important cause of the respiratory failure in such babies. Usually, surfactant synthesis starts from 24 weeks onwards and get completed by 28 to 30 weeks. Okay? So, if they suspect any preterm deliveries, then what they do is they give the mother the glucocorticoid injection so that it will hasten the maturation of the surfactant, so that the baby is not suffering from this respiratory distress. This particular syndrome is called as respiratory distress syndrome of the newborn or hyaline membrane disease. Coming to the last segment, that is work of breathing. In order to inflate the alveoli, we need to overcome the resistance offered by the surface tension of the alveoli. We need to overcome the resistance offered by the lung parenchymal tissues, that is elastic and the collagen tissues. We need to overcome the pleural fluid and its pressures, and we need to overcome the chest wall elastic recoil. All these requires work to be done. Okay? So, that work is actually required for breathing to happen normally. Okay? So, this is actually to overcome the resistance. There are two types of resistance. One is the tissue resistance, the other one is the airway resistance. Tissue resistance is offered by the elastic and the viscous resistance, whereas airway resistance is offered by the friction between the gas molecules themselves as well as between the walls. Okay? So, if we can calculate the total work done by the respiratory muscles during quiet breathing includes the work done to overcome the elastic resistance is 65%, the work done to overcome the viscous resistance is 7%, and by the airway resistance is 28%. Okay? So, this is the work done for the normal breathing. It will be much greater during exercise. To sum up, we have seen the muscles involved in breathing. The active inspiratory process is brought about by the diaphragm and the external intercostal muscles, helped by the accessory muscles during the needs. The expiration, being the passive process, is predominantly brought about by the elastic recoiling and also with the help of internal intercostal muscles and other accessory muscles when there is a need. Okay? And we saw about the movements of the respiration, chiefly the vertical movement of the diaphragm and the pump handle movement brought about by the external intercostal muscles of the upper ribs, and bucket handle movement brought about by the external intercostals of the lower ribs. Okay? And the pressure changes we saw, the pleural pressure, intrapleural pressure, which was actually negative, became more negative during inspiration and became less negative during expiration. Similarly, the alveolar pressure, which was 0 before inspiration, became -1, that is negative, and became +1 during expiration. Okay? So, that concept of transpulmonary pressure also we discussed, which actually reflected the elastic recoiling of the lungs. So, whenever the pressure became more negative, there was an increment or there was inflation of around 0.5 liters of air into the lungs during inflation, and same 0.5 liter of air was lost during expiration. Then we saw about the concept of compliance and elastance, wherein we saw when we measured the pressure and the volume and we plotted, we saw that During inspiration, it required more pressure to increase the volume inside the lungs, whereas during deflation, it required less pressure. So that the concept of hysteresis was evolved. That is, the lagging behind of inspiratory compliance and the expiratory compliance was discussed. We also saw about the alveolar surface tension force, which is brought about by the fluid that is lining the inner wall of the alveoli, which made the alveoli to collapse into the lungs, okay. And the role of surfactant in decreasing the surface tension, especially during expiration, the surface tension is much reduced than when compared to the inspiration. Finally, we saw about the work of breathing, that is the work done to overcome the resistance, namely the elastic resistance, viscous resistance and airway resistance, so that the required amount of air is flowing into the lungs, which will then be exchanged for oxygen and carbon dioxide. So that ends this mechanics of respiration. I thank our Chettinad University and NPTEL for giving me this opportunity to be a part of this online course on PFT. Thank you.